So, hello everyone. Welcome to this SNEV special series, and thank you for joining us. My name is Deborah Freire, and I'm a postdoc at the University of Minho in Portugal. So today I have the pleasure of moderating this session alongside my colleague, Lucien-Matteoni, a PhD student from the University of Sydney. So it is our great pleasure and honor to introduce our special guest, the Dr. Fabrice Lucien. He's an assistant professor of urology and immunology at the Mayo Clinic in Rochester, in Minnesota. He earned his PhD in immunology from the University of Sherbrooke in Canada. And in 2020, Dr. Lucien established his independent research lab focusing on elucidating the role of tumor-derived disease in immunosuppression as well as resistance to therapy. Additionally, he also leads a translational research program that is aimed at discovering and validating extracellular vesicle-based biomarkers for urological cancers. He also serves as the co-chair as the International Society for Extracellular Vesicles Blood Task Force, as well as he contributes to the organization of the latest ISEV Annual Meeting in Puerto Rico. So I kindly ask the audience to submit their questions in the chat box. These will be addressed at the end of the session. And Dr. Lucien, thank you once again for accepting our invitation. So we are thrilled about your talk and excited to hear your insights. You may now begin your presentation.
Thank you, Deborah. Thank you very much for the introduction. Sorry, I'm a bit getting out of a bit of sickness, so sorry if my voice is struggling a little. But yeah, no, I'm glad to spend some time a bit with you. For those who attended the education day at the annual meeting, you may found some redundancy because my presentation is pretty much the same. I just added a few slides and thoughts to share with you, but feel free to ask questions during the presentation. It shouldn't be too long. I expect to have about 30 minutes, but I want to leave more time for questions and any point of discussion that you would like to have. So yeah, let's start this off. So the ISEV 2026. So we had two portion of the meeting. One that is on an education day, the one day on the day before the start of the annual meeting. So we were super excited to host the meeting in Puerto Rico in San Juan. We had a beautiful convention center, obviously beautiful weather. For myself coming from Minnesota, that was quite a good change. So yeah, very nice locations, great staff, and I think overall we had some very positive feedback from the attendees, the sponsors. So as of now, I think we had a successful meeting. So we started with the education day, and one of your colleagues, Alexi Murillo, has been instrumental in organizing this day with Irina Matei. We had about, I think, 250, 300 attendees during the education day, which is pretty good. This is roughly 40% of the total number of attendees for the entire annual meeting. And we have different sessions that really covered, I would say, not just basic, but basic and some of advanced knowledge for EV research. So we had sessions on the fundamentals of EVs, the technologies, applications and clinical translation, rigor, reproducibility, and standards. And my presentation will focus a bit more on that. And we also have some viewpoints from scientists with two panel discussions organized during the day and some sponsored demo corners. So very dynamic sessions and a lot to do during this day. So very nice. On the annual meeting side, so we had about 700 attendees. We had 4 excellent keynote speakers that were selected based on some specific expertise that we think was valuable for the audience. We have Dr. Catherine Whitehead from Carnegie Mellon University, who is an expert in lipid nanoparticle engineering for drug delivery. Dr. Marka Woben from Netherlands, who discussed the immunomodulatory role of milk-derived EVs, in particular during breastfeeding. Dr. Weggio was well known for his work on understanding the mechanism that the tumor-derived extravesicles use to mediate local and systemic immunity. And we ended up, the last keynote speaker on the Sunday was Erding Sesgin from Karolinska Institute in Sweden, who has very unique expertise on the membrane biophysics and how the dynamics of lipids in a membrane can drive T-cell immunity, but also extravesical function and communication with recipient cells. We also had throughout the meeting 8 featured speakers that were selected based on top publications in 2025. So they were invited to present during the keynote sessions. And we had, after 21 sessions with oral talks that were selected from top scoring abstract, and that cover a variety of topics from basic biology of extravesicles, EV isolation characterizations, biomarkers, drug delivery, and so on. So a lot of great research presented during the meeting. And also we had some satellite symposium, mostly organized by the task forces and working groups to, again, develop initiatives, share progress on how to improve EV research in general. We had 2 poster sessions during the meeting. I think we had about like 800 posters. Sponsor lunches that allow individuals, attendees, to learn more about the technology that are being developed, and also some hands-on demo. And we have an amazing networking event on the Friday night on the terrace of the convention center. So very nice annual meeting. Very happy about how it turned out. So now I'm going to talk. Sorry, I'm going to talk a bit more on the presentation that I gave on the education day. So, I was asked to give my opinion or a perspective of what did we accomplish in EV research over the past 40 years, and how do I see, or how do we see the future of EV research. And I'd like to start with this Gardner style chart that maybe some of you know. So this is a type of chart that are used when during the development of novel technologies, so biomedical technologies, for example, when we evaluate what's the current status of those technologies and how do we expect the future and the potential translation for deployment in a clinic settings as a biomarker, for example, or again, like a new therapy. And so, we can divide this journey of EV research through this Gardner style view in 5 phases that I'm going to go over one by one. And I also indicated some of the time frame that I think represent those different phases. Obviously, this is subjective in a sense that, this is what I think personally those phases were up to now. So we start usually with like what we call the innovation trigger, which is really like the foundational discoveries of EVs that occur probably starting in the 1970s when extravesicle were for the first time sort of identified until like maybe 2005. So what are those key events that built the EV field? So we have the recognitions that EVs are biologically relevant entities, notably through their role as mediator of intercellular communication. And also one key event was the fact that we can detect disease-derived EVs in human biofluids, which opened the door for the development of non-invasive biomarkers, which we all know can have great benefit for public health. And also at the same time, we have the expansion of tools that allow for EV isolation, characterization, and also the cargo analysis. So now if we looked at the publication rates using the term extravesicles or exosomes, and using the PubMed database, you can appreciate that up to 2000, there was only 33 publications with those terms. And we see a great increase of this number of publications, reaching about 7,000 by 2020, And just between 2020 to 2026, we had more than 18,000 publications related to extracellular vesicles. Importantly, 20%, significant numbers, 23% of those publications were classified as review. So we see that kind of like innovation trigger and also this kind of rise of expectations just through like the numbers of publications related to the EV field. And so what did we expect from this initial research? Well, first thing was that EVs could be universal biomarkers and be used as clinical test for disease diagnosis, prognosis, prediction of treatment response, and really like across disease type. It could be cancers, it could be chronic kidney disease, or some even like autoimmune and neurodegenerative disorders. We were also expecting that the EVs are optimal therapeutic delivery vehicles, and so we were hoping that this would lead to a rapid translations to diagnostics and therapeutics. And so following this, we had an explosion of publications, enthusiasm from the research community, the industry sector, as well as the funding agencies. And of course, the commercial activity followed that enthusiasm. So this is where we kind of like reached the peak of inflated expectation for or so, because we had like a couple of markers of intense confidence that demonstrated the if I can say like the hype, the good hype of EV research. So EVs were marketed as the next major platform for liquid biopsy diagnostics, with the potential of overcoming some of the challenges that we see right now with other type of liquid biopsies, such as circulating tumor cells or circulating tumor DNA. There was a growing development of drug delivery and vaccine platforms. As of now, I think we have more than 400 EV-related clinical trials that have been completed or ongoing. And we also have significant numbers of EV-focused startups that have been created. I indicated the names of some of the most known, such as Kodiak Biosciences, Exosome Diagnostics, Exopharm, or again, Evox Therapeutics. There was more than 400 millions that were raised to develop those startups. And we also see some funding agencies to allocate some envelopes to fund EV research specifically, such as the NIH Extracellular RNA Communication Program. But we ended up, going through a phase called through of disillusionment. And so really the question was, what slowed the progress going from the discovery to translation into the clinic? But we realized as a community that there is a biological complexity that was maybe underestimated. And we're going to give more details about that in the next few slides. But just the heterogeneity and the size range of the EVs are already like two significant factors that sort of define the biological complexity of the EVs. We also make some mechanistic assumptions and novelty-driven claims that were not necessarily well established and not validated before we move, let's say, closer to the development of a test or a new drug. The pre-analytics that also, when we talk about pre-analytics, so it's really for example, biofluid collection, processing, storage, EV isolations. So those pre-analytics were mostly treated as technical details, but more and more we realized that they can really have a significant effect on the biological readouts. And then, this also was translated by, or at least, we have also evidence that the translation of some of the products advance faster than reporting quality. And I like to use the example of the dermocosmetics. Some of you know that, there's celebrities, influencers who have pushed the benefit of using extra vesicles from different sources, such as plants, to promote, anti-aging properties or anti-inflammatory properties. But we don't necessarily have the mechanistic basis of clear evidence of their benefit. And so this is where, like, the research has been facing some of the challenges, trying to, improve the understanding of the biology of extra vesicles, but also seeing a race to translate some of the initial discovery into the clinic before we have some true validation. And so what happened? What have been the consequences? But we had, found that, there's a limited clinical validation of EV biomarkers. So there was a study published in 2022 which showed that, there was more than 1,000 papers who have claimed to have discovered the next EV biomarkers, or at least finding signatures that could potentially change, the therapeutic landscape or treat or the management of patients with disease. Unfortunately, we have, as of today, maybe there's only three sort of biomarker signatures that reach the clinical validation. And there's actually one that, at least in the US, has been FDA approved as of now for clinical testing. So that just demonstrates, the sort of the as a reality check that, we had a lot of biomarkers that failed to move from the discovery stage to the validation stage. And there is also a commercial reality check, some startup expectations that have been proved to be premature. As an example, Kodiak Biosciences, which has been the first EV-focused startup, has filed for bankruptcy in 2021. The ExoDX prostate test that was developed by Exosome Diagnostic and acquired by Biotechne, the acquisition was about of $250 million, but that test was sold, I think, last year for like $15 million. So just this kind of highlight that, we need to do more work to understand not only the biology of the EVs, but to have also more realistic expectation of what we can achieve in the short term as well as the long term. So we could think that now, with this more pessimistic view of where we are with the EV field, that we have a problem in our field. But this is not just an EV problem. It's a typical pattern that you could see in biomedical innovations. And I can refer similar stories for stem cell therapy, microbiome therapy, even in the field of liquid biopsy. When something is discovered, come new, there is some commercial attractions, right? We see that we see the potential of commercialization, and of course, the economical benefit of having such discoveries going into the clinic. But after, there's some of we're going through this path of disillusionment and realize the complexity and go back to the drawing board to potentially bring newer strategies that would have a highest likelihood of success to translate into the clinic. And so, to support sort of those facts, there's some studies that have been done in the past that have been cited like many times, who kind of highlight what are the reasons why we struggle to reproduce or some of the preclinical data, and why it's so difficult to translate into a clinically meaningful test or drugs. And so in this paper published in 2015, it was estimated that about 50% of the spending done in preclinical research leads to irreproducible data. And the main reason is because we have a lack of reference materials that are necessary to benchmark, for example, or EV isolation methods, or again, some reference materials to evaluate the quality of biofluids that have been processed and stored for biomarker research. So those are just two examples. Study designs, importantly, data analysis and reporting, and I'm going to talk a bit more about that, and the laboratory protocols. And so importantly, when there was a paper published in Nature, when they integrated more than 1,500 scientists, asking them what are the factors that contribute to irreproducible research, and some of the key factors were selective reporting, methods, coded available, and variability of standard agents. So going back a bit to what was published in 2015. And that's something that I'm going to mention a couple of times in terms of the reporting is in the EV field, it could be difficult to understand how a specific research group has collected specimen, processed specimens, stored, isolate EVs, validated the yield, the purity, because sometimes the material method is not extensively detailed, or because we have some key information that are lacking to be able to cross-validate the studies. And again, this is not specific to the EV research field, right? This is overall in preclinical research, some of the key factors that we need to address to really make sure that we can move with great success in our discoveries. And so what was interesting is that instead of shutting down, the EV field responded with a cultural shift. And that was, I think, what described or at least illustrate the spirit of collegiality and also the collaborations between the EV scientists to really move the field forward, not just working in silo, but really doing this as a community. And so that's where ISEV established, the scientific reproducibility subcommittee that kind of govern, task forces, inter-society working groups, and also a committee on education and disseminations. And all together, those committees works on different outputs, establishing core standards such as the ISEV guidelines, which I think is a bit our Bible that, as a newer research group on EV research, or even as expert with many years of experience, those ISEV guidelines kind of set the backbone on, how to do reproducible EV research. And after the task forces and the different working groups, they work more towards biofluid, tissue-specific guidance, technology-specific harmonization, reference materials for reproducibility, as well as clinical responsibility by understanding a bit more the regulatory requirements from the federal administration, but also establishing statement to allow for to provide guidance on how to translate our discovery into a clinical essay. And so this just illustrates that the field did not retreat from complexity, but instead built an infrastructure to address it. And I think over the past 5 years, we saw some significant shift in the way the EV research is done, and also that is translated by the publications, and also the move to the new phase of the cycle when we start to see more translation of our discovery closer to the clinic. And so this is where we reach the slope of enlightenment, which I think started about 2020, and we are right now in that slope of enlightenment to potentially one day reach the plateau of productivity. And so here is what I think 5 shifts that help move the field into the right directions, into that phase of enlightenment. So one is moving from isolation language to enrichment and residual uncertainty. So I think, if you look at different talks and also some of the major initiatives that the task forces are doing, when we work with extracellular vesicles, it's important, regardless of the source, to assess using benchmark methods the yield, the recovery, and the purity. And this is also important to understand those definitions and how can we measure them in a reproducible manner. And I think it may seem very simple as a term, but I think those terms are used interchangeably. And this is, I think, inaccurate, because those 3, yield, recovery, and purity, have different meanings and could have different impact on your downstream analysis. We also want to move from single readout to orthogonal characterization. We saw at the ISEV a variety of technologies that allow for characterizing EVs from immunoassays, flow cytometry, NTA, advanced NTA, and so on. So I think now we have access to more platforms that allow us to validate or test or analyze our EV preparation, not just on one platform, but on multiple platforms to have a higher confidence on what do we work with. And so those orthogonal characterization allows to define the EV identity, the abundance, or again, like I mentioned, the purity. Also, we really want to push more on the reporting. The pre-analytics determine the data interpretability and enable cross-validation studies. It's important to understand how the blood is processed to generate plasma. And, as part of the blood task force, this is something that we work heavily with our group. But for example, residual platelet in plasma is a major contaminant for downstream analysis of RNA and proteins. And so depending the way you process your blood, you could have residual platelets that could differ from 10 or 100 times between two samples. And this is something that is important to report because it would influence the downstream results and the data interpretation. And so as of today, we maybe don't know what is the gold standard for isolating EVs, but what we can do instead is reporting the way we process the EVs, so we can generate data that in few years we can analyze in meta-analysis and figure out maybe what are the steps that are detrimental for high quality EV samples versus what are the key factors that are really going to help to generate some high quality EV preparation. Also another shift that is important is moving from descriptive association to mechanism-aware studies. So really understanding, if the effects that we see biologically are EV-associated, or maybe, having some co-isolated molecules that are carried by the EVs that can actually participate in those effects. And this, can be addressed by using proper controls, proper experimental design. And I would say keeping like a critical mind that, we cannot isolate pure EVs with perfect purity. And so there's always going to be some confounding factors that could play a big role and have a significant biological effect and be relevant for your studies. But this is important that, we consider those different limitations in EV isolations and analysis when we do our studies, and to not have some non-validated claim. And finally, in the context of clinical translation, we want to move from publishable signal to decision-grade evidence. In the development of biomarkers and therapeutics, we need to understand, does my research really going to change scientific decision, clinical decision, or even regulatory? How can I really make a direct impact to public health by just, designing a study that is aligned with what is needed? And so if we embrace those 5 shifts, we will move with what I think the plateau of productivity that is summarized, I would say, in like 3 categories. One is reproducible biologies. So we want to come to a phase where we can replicate findings across labs, or even, invalidate findings, by, multiple ways. It's I think, as a broad research field, we tend to neglect, negative data, but negative data sometimes could be good data to share. My own experience is that, I learn a lot from some of the negative data that we generate, and sometimes more than the positive data, because it helps you to refine your hypothesis and ultimately leads to greater discoveries. We also need studies that can distinguish EV-associated signal from co-isolated confounders, like I mentioned, and also, allowing comparison of methods and metadata, not just local success. I want my research to be validated by external groups. That would further support the validity and also the relevance of what we're doing. And we hope also that the journals focused on EV research, such as the ISEV journals, EVCNA, and others, will support open science frameworks sorry, and standardized reporting to, again, allow cross-validation studies. We want to develop clinically useful tools that could be biomarkers, assays, and also benchmark the performance against the current standard of care. And finally, we want some regulatory credible products, especially in the context of therapeutics, with better defined source materials, QC potency, and also manufacturing protocols. We want to understand a bit more the mechanism and the doses are interpretable enough for translation. For those who work with in vivo research, I mean, in my field of cancer research, in vivo studies is, sometimes difficult to interpret in the context of, is it something really that happens in vivo in patients? Does the dose that I inject IV in my mouse tumor model, is reproducible of what we would find in a patient? So those are like questions that we need to address so we can make sure that our discoveries have some potential clinical impact. So I would say that, EV research is poised to enhance our understanding of human biology that can deliver real public health benefit. I think I like to say that, the future is not just novelty, is also credibility. And I think we can achieve it by biological depth, analytical rigor, and transparent reporting. And finally, I would say that the future of EV research is not less ambition, is just ambition that can survive scrutiny. And I think, the ISEV 2026 has been another illustrations that we're moving in the right direction. And, I'm very much looking forward for the future. And for those who don't know, you are all invited to join the ISEV 2027, which will be held in Busan, Korea, from May 4 to May 7. So hopefully you make the trip. So I thank you for your attention, and I will take any questions from the group. Thank you.
Thank you very much. That was a wonderful talk and well-rounded. Please enter any questions that you have in the chat box or feel free to unmute yourself as well. That was wonderful. I might get started. You mentioned now this like peak of enlightenment. There's many things we can do, and you listed the 5. I just wanted to ask how long, so from here on, when do you think we can deliver the clinical promise from the EV world? Like, would it be a decade or 5 years, in your opinion? And what are, like, if you had to pick out one problem that we must solve to deliver this clinical promise, would it be like standardization, or do you have a different opinion?
Yeah, no, I mean, this is just these questions, we could spend an hour on it, obviously, and it's going to be a subjective answer, but I would say that we already see some very promising movement in translating some of the discovery into the clinic from a biomarker perspective and also from a therapeutic perspective. I think on the biomarker side, we had a chance to have Dr. Tumi Getush, who gave a talk during one of the Cytedite symposium at the ISEV, who is working on ovarian cancer test using EVs, so detecting some of the biomarkers on the surface of the EVs in ovarian cancer. And it has been very inspiring talk to understand what is needed to reach the clinic. Because, when you discover your biomarkers, in a retrospective study cohort, so, you're going to design your experiment, you're going to have a discovery phase, and then you realize that you have some protein genes that seems to have some diagnostic potential. So you need to understand, are they actionable in a sense that, are they addressing a significant unmet need in the field? And for that, I think my own experience, because I'm also working in this area, is you need to partner with clinicians. I think, there's some research groups who are working on biomarker discovery. Without having no idea of what is right now the standard of care or what is needed in the field. I think this is something that I learned by sitting down with clinicians and asking them, what is your problem in the clinic? And something also that you realize is that, I work in the field of urology, and you can talk to 10 different urologists that treat prostate cancer. And the 10 would have slightly different approach to treat patients. It's not a binary, like there's a standard of care, but there's also a large gray area because each patient is different. So it's really understanding the clinical unmet need, do the discovery, and then how to move the discovery into the more the validations. But I think the big challenge over the past few years was what techniques can we use, right, to move the EVs into the clinic. And again, I'm going to refer to some very, common thoughts is, for example, we all use ultracentrifugation to isolate EVs. What about if tomorrow I bring a cohort of 300 patients? Can we implement ultracentrifugation to isolate EVs before detection? Maybe not, maybe yes, I don't know. We want something that is clinically scalable and implementable. So now we think about what other techniques can I use? And so I think in the past, we were limited in the techniques that were available, and there was also limited knowledge on, their advantages and their limitations. Now I think, we're getting more and more reference materials, standardized methodology, calibrated methodology that allow orthogonal validation. So this is, we start to, understand a bit more what we isolate, what is the composition of our sample, can I reproduce that isolations, that detections, and so now we can move toward the validations and kind of move forward closer to the clinic. In a therapeutic world, this is the same thing, right? It's like, can I control the way I isolate EVs? Can I control the way I characterize the EVs? And do I establish the right models to study their therapeutic value? And I think, there's some very interesting clinical trials, like I said, about the dermocosmetics, but, here at Mayo Clinic where I work, there is some clinical trials on, using EVs for tissue regenerations after, surgery, or again, for tissue after tissue scarring. And the data are very interesting. Now, are the effects really EV derived because you have put EVs on top of the tissue? Again, there is some mechanistics to do, but I think, the field is moving forward. What is important is that the community accept and understand the current limitations and maybe some of the fail, experiment that we had, and really, promote, embrace transparent reporting, validation studies. And, I think it won't take 15 years to have something in the clinic. Hopefully not, I hope myself to have something earlier than that, but probably in the next 5 years. But no, I think so yeah. So I think, it's again moving in the right directions. We have evidence that there is some technologies that are being that move closer to the clinic. But time will tell. But this is, as a community, the efforts we can make to make it happen.
Thank you. Just a follow-up question before I move on to the chat. So do you think we are under-investing as a general in EV field is under-investing in basic biology of extracellular vesicles and moving more towards the rush of omics and biomarker discovery, or do you think they go in
Yeah, I
parallel?
mean it's good. It's good. I mean, and especially now with if we think about the increasing use of AI, for example, for data integration and analysis, which I think is, making incredible, I mean, it's revolutionizing the field. I mean, again, yesterday I was chatting with some of my lab members saying that, 3 years ago, I wasn't using the word AI, and now it's every day. The good news is that for example, the artificial intelligence will help us maybe to start asking questions that we were not able to do before, because now we have the computing power to generate some answers to complex questions. Now, in terms of the basic biology, I think it's just there's always going to be, a race between. It's not supposed to be a race, but there's going to be a race between, trying to translate a discovery into a commercializable product. And I think, we have to again acknowledge, like I said, the complexity of the biology of the EVs. The EVs won't solve everything. And you take the research, even like I said, in other field, like stem cell therapy. How many times we heard about cardiac stem cells to regenerate heart, right? And I think there's now data more and more that are being generated that are maybe more promising. But 15 years ago, 20 years ago, when they start talking about it, a lot of the data were not reproducible because it's more complex than we thought. We need to accept that. So I think even if the funding situation is complicated, I think we have some funding that support, basic biology research, and I think we need it to understand, the role of EVs for intercellular communications, but also as a mechanism for the cells to, release some waste cellular products. So I think, when you look at the ISEV, even if the ISEV meeting, the theme was EVs in translation, that was something we decided to use because we want to promote, that kind of rigor and reproducibility mindset to really move toward the clinic. But, a lot of the sessions were still focusing on the fundamental biology. We need that. It doesn't work independently. You need to understand the biology to be able to design the next generation therapies and also drugs. So yeah.
Thank you. That was great. I will just read out the question from the chat. This one's from Sarah. Looking 5 years ago now, and 5 years from now, what do you think is the most consistent challenge in the EV field, and what do you consider the biggest effort being put in to address that challenge, which you kind of touched on?
That's a good point. Yes, again, I consider myself as still a young investigator. We're not that far from each other. So it would be interesting to have that question with someone who has 20 years of research. But what I saw, because I started EV research in 2016. So it's actually 10 years now, I realize. But it is, I think every time that I was going to ISEV and attending those satellite events, my first satellite event was about EV flow cytometry, because this is a technique that we use a lot in the lab, and we're proud to say that we really acknowledge what the experts in the field were doing and asking us to do to make sure that our data are reproducible. And I think the EV flow cytometry field has made some significant progress thanks to individuals like Joshua Welch, Edwin van der Poel, and Andre Gorgens, who are Vera Tang, Jennifer Jones, who led the field forward. And I think my lab, for example, has EV flow cytometry tests in clinical validations right now, and that has been possible because we follow the guidelines and the recommendation of those experts. And when I studied EV flow cytometry, I didn't know anything about flow cytometry, and so we started with something very difficult, and we understood what was the current limitation, and we decided to address them right away. And I think that's the reason why I think we made so much progress in the past few years. So I think what I saw over the years was always report. That's something that is really key. All of you that work on EVs, you know that you can centrifuge a sample at 10,000 G and centrifuge another sample at 20,000 G, and that can make some significant differences. If we don't know exactly the way the samples are processed, it would be very difficult to interpret the data. And as a scientist, I need your data. I need to read your papers and to understand how you did, so I can be inspired and build my research projects along those findings. So I think, the challenges that we faced was, a lack of standardized methods to isolate and characterize EVs, a lack of reference materials, and a lack of reporting. And so I think that, we should all, as a community, move forward by doing that. It doesn't take long to have a supplementary document that lists in great details your experimental design and what methods you have used. Something that we like to do in the lab, and I saw that in other groups, is every time that we do experiment on a specific projects and that we realize that it didn't work, like it failed, we add those data in supplementary informations, because sometimes you can say, oh yeah, I'm comparing those different temperature for EV labeling, but I don't know if they really have an influence or something like that. And if we see that, for example, they have no influence or they have a significant influence, but maybe it's not important for the conclusion of our papers, we still deliver those data in supplementary material, because I think it could serve anyone who will be reading the paper. So I think, this is what I think the biggest effort that we should do is, report, standardize the methods, the main methods that you use, standardize them, and kind of establish some reference samples that you can use, across different studies, especially when you optimize technique. So to give you an example, we work a lot with blood and urine. Every time that we optimize a method, we spike our blood and urine with recombinant EVs, like fluorescently labeled or so on, and we freeze that. And we always use those reference materials that we generated in the lab to standardize our methods. And we found, some consistency that help us to really tailor some methods specifically to address some of the questions that we have. So it doesn't take much effort or like a significant knowledge to establish those standards. So yeah.
Great. Thank you. Next one's from Carlos. He says, I'd like to hear your thoughts on potential impact of single EV characterization methodologies in helping the field. Do you think these approaches could play a major role in accelerating clinical translation?
Yeah, that's a good question. I'm a bit biased already, because I'm a strong believer of the single EV technology. I think yes. I think they have a very strong role in clinical translation. So again, I'm just going to refer a bit of my own experience, because I'm more comfortable with sharing this with you. My lab work a lot on method development and biomarker discovery for urology cancers. One of the methods that we really love to use is EV flow cytometry. And so I think in prostate cancer, we have been one of the first to establish that the circulating level of prostate cancer-derived extravesicles correlate with the tumor burden. So, if you have a patient with healthy disease, healthy control, localized disease, recurrent disease, metastatic disease, we see a beautiful increase of the level of circulating EVs. And we, make the claim that the levels of EVs that circulate in the blood are representative of the tumor burden in a patient. And I think as of now, this is by using single EV characterization method that we have been able to make that claim. That doesn't mean that non-single EV characterization methods do not have a potential. I think in the context of discovery and validations, the proteomics, RNA sequencing, immunoassays, they have some strong potential, as long as that it's demonstrated that you have some sort of correlations between EV concentration and your readout. So I think this is something that is very important. Now, I think what is beautiful with the single EV characterizations, and you can talk about flow cytometry, but also SP-IRIS, so like the Leprechaun system, even like some of the more like microscopy-based method, when you can quantify EVs at the single particle level. What I like is that in some cases, you don't necessarily need any isolation or pre-purification of the EVs, which I think is great because, in our case, we work directly from plasma and urine. We don't do centrifugation. We do minimal centrifugation, but we don't use extensive or labor-intensive method like ultracentrifugation, SEC, to measure the signal. That doesn't mean that we have a perfect signal. We acknowledge that we have, debris, co-isolated molecules, background noise. But in our context, what we measured is not to influence, or at least we're able to capture a signal that is not too affected by the composition of the sample. Now, if we were doing, discovery, maybe we will use a method when we have, more careful enrichment of the EVs. So I think the single EV characterization has a lot of potential. And I guess that, over the next few years, we're also going to have an increase of the type of platform that can be used. I know that there is a lot of microfluidic-based devices that are being developed for capture and detection of the EVs. And I think those, when they're going to be accessible for, the research community like us that are not, experts in microfluidics, I think that's going to propel the field forward. So, yeah.
Thank you. I think the next question kind of follows up on your last statement. What are some tools that you wish to have or the field should develop to advance EV research? So is it more single EV?
Yeah, I mean that's a good question. Well, I mean, I think the reference material is the one thing, right? So if you take, for example, we don't all have access to the state-of-the-art technologies, right? To use the EVs, it depends on the infrastructure that you have access. But I think as a new investigator, when you start doing the EVs, and let's say you get access to NTA, you get access to MRPS, you get access to flow. I would say at first it's very intimidating because I think we all realize how when you start using the instrument, how variable the data can be, right? And sometimes you cannot necessarily explain them. And I think without some reference material that help you not only understand the technology, optimize it, and kind of like standardize to make sure that your data are reproducible over time, I think this could be very difficult, or at least you're going to have some slow progress at the beginning. So I think that we should have more reference material. The reference materials are not necessarily developed by the industry sector, right? I mean, you look at some of the work from Dr. Anne Hendricks, Dr. Charles Lai, for example, Dr. Charles Lai has developed, they call it the palm grid system. So this is a reporter that is incorporated into the EVs with the luciferase, and GFP, and you can use that to track your EVs that you inject in vivo. This started from like a wet lab research, and I think now a lot of people are using it to track EVs in vivo. But you can also use that as a reference materials to standardize your method and say, okay, does my flow cytometry detect EVs? What size do I detect EVs? What fluorescence? And when you change your isolation method, how does it impact on your EV isolation and detection? So I think reference materials is really something that we need to push forward to make sure that our methods are reproducible. And also that I give you a very simple example that I think it's kind of summarize a bit what we need. Flow cytometry. Nowadays when you run in the past, when you run flow cytometry, people report the concentration on the number of EVs that they detect. But we have now today's, I don't know, like 8-9 different flow cytometers that can detect EVs. How do I know that we detect the same size, the same concentration? What we want is that if I take a sample and I decide to measure, let's say, CD9 positive EVs in plasma, and I detect, I don't know, 10 million CD9 positive EVs per mil, and I'll send you that sample. I want you to detect 10 million CD9 positive EVs per mil. But just that simple experiment, I can tell you, will be very difficult to perform without having standardized and calibrated instruments. And there's a work that been recently published by Dr. Bettin and from Dr. Newland and Van der Poel's lab, like I think it was last year, who showed that using standardized and calibrated flow cytometry, you can significantly reduce the variations in measuring EVs across international labs. So I think this is direct evidence of the tools and the sort of the actions that we need to take to advance the field forward.
Thank you. Now for the question from Kirill. Two questions. One, is there any EV-specific assay that can be named as a successful example? And the second question is there any true disease-specific EV biomarkers?
Okay, good question. So first question, EV-specific assay. So if we talk about, so I will need to understand if it's meant like clinically used or more in what context, basically. Maybe Kirill, if you want to refine your question. But I'm going to start on this with the second question, and maybe you can clarify on the first one if it's more like what is clinically used right now or not. In terms of disease-specific EV biomarkers, well, this is, I think, the questions that we all want to answer in the biomarker research field. I think something I gave a talk in the past on EV biomarkers. Something that we have to take into consideration is that why cell-free DNA? If we take the example of cell-free DNA, circulating tumor DNA, why circulating tumor DNA today is making some major breakthrough in the field of biomarker research versus the EVs? Well, one, ctDNA was discovered before EVs. Second, they have, I say they because I don't work on it, they have calibrated technologies like sequencing platforms, isolation of DNA from biofluids. It's standardized. There are kits, commercial kits, validated. We know how to measure DNA after isolations. We know how to measure purity, and we know how to sequence DNA and to analyze the data. The analytical pipeline might be a bit more variable, but I think we know much more how to handle DNA. Now, the other aspect is that ctDNA, initially, when you measure ctDNA as a marker of tumor burden, but it's almost binary. You detect mutations or you don't detect mutations, right? If you detect mutation in a patient, theoretically, there is cancer. You shouldn't have, some of the somatic mutation, in non-tumor, in tumor-free patients. When you look at the EVs, what do we do in the EVs? The EVs, we mostly measured RNA and protein cargo, or lipids, or metabolites. We measure RNA and protein, but RNA protein, for the most part, are not specific to a tissue. They can be associated, meaning they express by a tissue. They can be enriched, meaning that you can find more of those RNA and protein in a specific tissue versus others. But there's very few RNA and protein that are uniquely present in one specific tissue, and even less in the tumor, because the tumors, yes, there is mutations, but the RNA and the protein profile is for the most part similar to a normal tissue. That's also for the reason why sometimes the immune system is not able to recognize the tumors, because it's kind of what we call like tolerance, is there's not enough differences between the tumor and the normal tissue to be able to be recognized by the immune system. So what happens when you do biomarker discovery? But now you try to measure RNA and protein, and that's something that if you already do that, you're maybe going to smile, but what do we do when we want to look for biomarkers? We're going to compare normal tissue, cancer tissue, we're going to look at RNA and proteins, and we're going to do beautiful volcano plots to say, those are my gene and protein that are twofold increase with a p-value less than 0.05. And so we hope that those genes and protein can serve as biomarkers. Now we try to translate that into the EVs. So you want to see in the EVs that the cancer patients has 2, 3, 4 times more of those RNA and protein versus the normal patients. But now when you realize the variability that can be introduced by the biofluid isolations, the EV isolations, and the characterizations, I can tell you that just by pipetting, you can introduce a twofold difference between your samples that is not biologically derived. So how do you assess that what you see is not technically derived, but more biological effect? And that's what I think is very difficult to translate biomarkers. I mean, we do it myself in the lab. You know, we have these biomarkers. Oh my God, it's 30% more increase in patients. 30%? If I just spin a bit differently my samples, I introduce 30% variability. And this is, I think, the reality. So the challenge is, you know, what is actionable? And if we decide to use RNA and proteins, we have to be 100% sure that the differences that you see between your cases and your control are biologically derived and not technical. And that's, I think, a challenge that we all trying to solve right now the best that we can. In terms of the EV specificity in clinical aspects, so there is one test called the ExoDx prostate test, which I think has been the first to reach FDA breakthrough designation device developed by Exosome Diagnostics, which was basically a total EV isolation from the urine and RT-qPCR or ddPCR, the PCR of three genes that are known to be elevated in prostate cancer, and they were using that to kind of like improve the diagnosis of prostate cancer pre-biopsy. There is, like I said, one from Mercy Bioanalytics. We're moving forward, the ovarian cancer test, also for diagnosis. And after, there is a few that I think, you know, we saw some interesting data, some startup that started, but ultimately that didn't really pan out into clinical validations. So I would say that, you know, right now, most of the tests are between discovery to analytical/clinical validations. So like I said, I think over the next 5-6 years, we're going to have maybe some positive news on some EV tests that are moving closer to the clinic. Oh, we also have, I remember, we also have the company Mersla Bio, who has a liver-specific EV assay that they have that is multi-omic, that I think also have recently received the FDA breakthrough designation device, but the clinical validation are still pending.
Okay. Thank you for the questions. And with that, we're on time to close the session. I would really like to thank Dr. Fabrice Lucien for this wonderful, wonderful talk. The meeting was truly great. And for those of you who didn't have a chance to attend, hopefully this session provided some key insights into the meeting. And I would also like to thank my co-moderator, Deborah, and all of you who attended today's talk. I hope you have a wonderful rest of your day. Thank you very much again.
Yeah, thank you. Thank you for the invitation, for the nice discussions. Have a good rest of your day.
That was great. Bye.
Bye.
Bye, everyone.